get rid of them? Why would you get rid of those? Yeah, so if you're gonna make transgenic corn, you probably don't want a field of corn plants with big tumors. So we could remove those hormone biosynthesis genes. What about the opine biosynthesis genes, so the bacteria food?
Keep it.
Keep it? Why would you keep it?
It's food source for the bacteria.
Do you want... So it's expensive for the plant to be making these opines. You don't necessarily want your plants to have a bacterial infection. But is it doing anything bad to the plant? No. Right. So that one could sort of go either. Could go either way. I would remove it. The less extra stuff you're putting in, the better. There's no need for it. It doesn't necessarily do anything bad, but I'm gonna take it out, right? Because you don't need to make bacteria happy. We want to make our corn plants happy. We want to make our corn plants that are very productive for humans. And these poor agrobacterium they don't need food. You kill them in the process of transformation anyway. So we don't need to keep them happy, unfortunately, for them. So now we've taken out everything that the agrobacterium had for their natural gene transfer system. So what would you want to put? What would you want to put in?
Gene that's resistant insects.
Great. So we're going to put in a gene of interest. So what are genes that are provide resistance to insects? Josh just said. So what are some other traits that you might want to put into plants besides so resistance to insects? What are some other choices? That was a great first choice.
Nutrients, even resistance to like pesticides and stuff like that.
Sure, so pesticide resistance. Anything else? Yeah. Herbicide resistance. Actually, this would probably be herbicide resistance. We'd actually want to put not resistance to pesticide, but you could put a pesticide gene in there, right? Could you put genes that are good for human nutrition in there? Nah.
Yeah.
Why would we want to put genes for human nutrition in there? That would be a silly thing to do, right? So,
Maybe something for a better color, so it looks good on the store shelf too. Like a nice deep yellow.
great. Commercial. Sure, say commercial looks or less browning. That one's been in the news lately. So things that make it look good. Yeah.
Higher yield.
Higher yield. So there's a lot of different things you could do when making these transgenic plants. You can help them be resistant to pests. You can help them be resistant to an herbicide that you want to spray on your field to kill weeds. You can have them make their own pesticides. You can think about human nutrition, commercial looks or traits, increased yield. So I'm going to sum up all of these things into just one. I'm gonna call it a gene of interest. So it would be the DNA sequence, that's the protein coding sequence for whatever trait, whatever gene you're interested in. So all of these things fall under this category of gene of interest. What else do you need to do? So thinking about the bacteria that you transformed last week, you needed a way to know what was transformed and what wasn't.
Markers.
So you want a marker gene. Right, so what you'll see when you look at your plates and what you see, the reality of this is that it's not 100%. Every cell that comes into contact with these agrobacteria doesn't automatically get transformed. The efficiency is actually fairly low, and so you need a way to select for your plant cells that have actually been transformed. So this is exactly what scientists did when they figured out how to use the system. Is they took that Agrobacterium ti plasmid, they made a couple of changes in between that left and the right border, that tDNA, the part that gets transformed into the plants, took out the things that they didn't want, and put in the things that they did. There we go.